everyone, I am back at the LTER. My name is Jennifer Jones. I'm a postdoc in Sarah Evans' lab. I'm interested in studying microbial ecology and how microbes influence ecosystem processes.
Hi, my name is Grant Falvo, and I'm a doctoral student here at the Kellogg Biological Station, and today we're at the main cropping systems experiment to show you how we're going to be using these Rainout shelters to do an experiment next summer. Many scientists, land managers, and policymakers are interested in how climate change-induced changes in the precipitation regime for this region are going to be affecting the different agricultural landscapes. And KBS is uniquely positioned to test not only how climate change is going to be affecting these different agricultural landscapes, but also to understand the underlying mechanisms of those responses and some potential intervention points to make these agricultural landscapes more resilient in the face of these disturbances.
So for this experiment, we decided to focus on carbon and diversity to building resistance and resilience to drought and agroecosystems. We use research at KBS to inform the decision to use carbon and diversity as treatments. In 2012, there was a drought at Kellogg Biological Station that decreased soybean yields. But in the no-till treatments, which have higher soil organic matter, the soybean was more drought tolerant than in the till treatment, which has lower soil organic matter. There's also been many studies at KBS that have shown that increases in diversity can increase resilience and resistance to drought. In this experiment, we will induce a 6-week growing season drought by blocking 100% of precipitation using Rainout shelters. We will do this experiment in 3 different agronomic treatments. First, a conventional tillage with conventional fertilizer inputs, a continuous no-till treatment with conventional fertilizer inputs, and an early successional field treatment. We will run this experiment for at least 3 years. In order to test the impact of drought legacy on crop yield, we will have 2 main treatments. The press treatment will have precipitation decreased via a shelter for 3 consecutive years. The pulse treatment will have precipitation decreased for 1 year.
All right, so underneath each of the Rainout shelters, there's going to be 3 subplot manipulations and 1 unmanipulated control. These subplot manipulations are designed to test different mechanisms of resilience in the different agricultural land uses. So in the conventional agricultural plots, where these plots are the most depleted in soil organic matter of all the agricultural plots, we're going to be testing adding organic matter as a mechanism of resilience. But within that, we're going to be testing 2 different versions. So the abiotic effects of organic matter are important to understand. So for that, we're using biochar, which is essentially metabolically inert to microbes. So we're going to be testing whether maybe, for instance, it holds on to more moisture during the drought and provides resilience to the plants or other things that way. So the second overarching category of resilience that we're going to be testing here has to do with how microbes metabolize different types of plant material. So in one subplot, we're going to be using sorghum sudan grass, which is relatively easy for microbes to degrade, and it's therefore is probably going to select for some fast-growing microbes with general metabolic capabilities. Whereas in another subplot, we're going to be using switchgrass, which has a lot lower nitrogen content, is therefore much harder to degrade, and therefore we're thinking it's going to select for microbes that have a much more broad metabolic set of capabilities. So this will show how the metabolic strategies of microorganisms may interact with their climate strategies, or how they're going to be responding to the drought and the rewetting, as well as show how these treatments are affecting insects, plants, and other ecosystem responses to the drought and rewetting.
In the no-till and early successional fields, where soil carbon storage is high, we wanted to test the impact of diversity on building resilience to drought in plants and soil. We initially tried to study diversity by increasing microbial diversity in the soil, but this can be really difficult. So last year I did a pilot experiment where I tried different application methods for microbial inocula aimed at increasing soil microbial diversity. At the end of the summer, I found out that none of the application methods that I tried increased soil microbial diversity, altered microbial community composition, or altered plant growth. Therefore, we decided to reduce diversity in the soil. We decided to use reduction treatments for 2 agriculturally relevant taxonomic groups, mycorrhizal fungi and nematodes. Mycorrhizal fungi are important plant root mutualists. They can increase access of plants to water in the soil and increase plant drought tolerance. Nematodes eat plant roots, fungi, and bacteria. And as they're important for decomposition, they're important for carbon and nutrient cycles in the soil.
So due to COVID-19, we had to postpone the Rainout shelter experiment until next summer, but this gives us some time to do an analogous experiment. So I'm taking some soils inside and doing a similar drought and rewetting with some plant and microbial manipulations, and this is going to provide us with some deeper mechanistic understandings that will inform how we interpret the Rainout shelter experiment next year and in years to come.
Because the Rainout shelter experiment was postponed, I'm using this year to focus on learning skills that I will need to do the experiment that I want to do next year. Next year, I want to study how drought and carbon availability influence how microbial communities build soil carbon. I will use a functional trait and trade-off based approach, in which I will use shotgun metagenomics to identify the abundances of microbial traits in soils. This trait-based approach will allow me to develop a predictive framework to understand how the response of microbial communities to drought impacts building soil carbon. These are just a few ways that this Rainout shelter experiment can benefit soil microbial ecology. But we hope that researchers will use this experiment to study plants, soil properties, invertebrates, and more. So stay tuned on our future research linking carbon and diversity to drought tolerance. See ya!